All right, why don't we kick off? Welcome everyone to our May installation of the Type 2 Diabetes Knowledge Portal workshop and webinar. It's great to see everyone. And today's webinar is meant to be an interactive demo. I'm very happy, Maria is I think very happy she gets to give a live demo, and there's many reasons why this is a rare treat. We will be previewing the beta release of our Common Metabolic Knowledge Portal. And we're going to be recording it today, so if you miss it, or you want to see it again, it'll be on our website going forward. Also, we're doing a little different function today. We want you to be able to ask questions as we go. So instead of the chat function, which you see at the bottom of your Zoom typically, you're going to see a Q&A. So if you post a question in there, we're going to stop and we're going to address it. Allie's going to be monitoring that. I think I got that right. I think we see the questions as we go, I think, but we'll be addressing those as we go. And we can also unmute lines, and you can unmute yourself, I believe, to ask questions. Is that right, Allie? How is that going in terms of muting?
Yep, we can unmute participants.
Okay, all right, so ask a question if you want, and then we'll unmute. So hopefully this will work well, and we'll get going. And I'll just do a quick intro, and Maria's gonna talk about, we're gonna have 2 topics for today. Let me flip to my slides and we'll get started. It's down, hopefully everyone can see that. All right. So today we have 2 topics. One is a very brief topic just to remind you of our current release that we had for the Type 2 Diabetes Knowledge Portal in terms of a massive influx of data that came online as of about a week ago, covering both diabetes complications, and cardiometabolic traits. So Maria's going to talk about that real quick. And then we're going to shift the rest of our time today for the beta release of the Common Metabolic Disease Knowledge Portal. We're going to talk a little bit about its roots. Just to remind you, many of you were probably here for the March webinar, where we talked a bit about this in more detail, but I just want to remind everybody of the origins of this knowledge portal and why we're coming at it this way. And then Maria is going to give a live demo of what we've built so far, and then talk about what's to come over the next couple months and our official rollout plan for when we plan to release this live and phase out the old sites. That's what's coming over the course of the summer. It's been very busy for our engineers and computational team, so we're really excited to share this with you today. And really, this is about feedback. The site is under active development, as you'll see, and we'll be adding to it as we go. And so we really want to enable you to have feedback and take part in the development process and give us feedback. So we're going to also show you ways that you can give us feedback actively on the site, both today and going forward. So with that, I'll turn it over to Maria for a second while she talks about our current release.
Yeah, so I'll just review briefly. I think this is probably our largest data release ever. I think it's 15 data sets, and this page lists data sets that have gone into the T2D knowledge portal, but all of them have also gone into the Common Metabolic Diseases knowledge portal that Noelle's going to talk about. So just to briefly talk about what we put in, there are some new data sets for glycemic and T2D complications. So the BioMe AMP T2D GWAS is a set of data that we've had for a while, and we've been repeatedly performing different analyses on it. And so the latest analyses looks at 5 different complications of diabetes that were ascertained in type 2 diabetics. So that's kind of cool. We've also loaded the data set from a recent paper from James Miggs and colleagues from the MAGIC consortium that looks at the change in fasting glucose levels in non-diabetics over time. And also we've loaded a diabetic retinopathy GWAS from Lucia Sobrin's lab that looks at retinopathy and a couple of its subtypes as well. On the cardiovascular side, we've loaded 2 data sets from new papers that have just come out. One of them just came out this morning, and the other came out a week or 2 ago with some measures of cardiac function. One of them looking at left ventricle MRI measurements, and the other one looking at PR interval EKG measurements. We've also got exome sequencing for the UK Biobank atrial fibrillation data set, and that is both single variant scores and gene level scores for atrial fibrillation associations. We've loaded a big hypertension exome chip analysis and also a huge meta-analysis of cardiogram plus 4CD and UK Biobank for coronary artery disease. On the anthropometric side, we loaded a large giant UK Biobank meta-analysis. And then we've, in the purple box, there are just some data sets that have multiple phenotypes, so they could fit into all these categories. One of them is from a local colleague of ours at the Broad, Joanne Cole, along with Jose Flores and Joel Hirschhorn, who did an analysis of 185 dietary habits and preferences and patterns from UK Biobank. So we've loaded 5 of these, some of the ones with the biggest sample size, into the knowledge portals, but you can also download the whole set of summary statistics from the portal. We've also loaded selected traits from FinGen, and from the last 2 studies, UK Biobank Mendelian Trait GWAS and 409K GWAS. These are GWAS studies that support a new method of predicting effector genes that we are currently working on with the researchers, Brent Richards and his group, and this will be available in the portal within the coming months. And so we just want to have the underlying data on hand before we implement that. And the last thing I want to mention is that many of these studies are available. The full summary statistics are available for download from the portal.
Beautiful.
On to you, Noelle.
Excellent. So I think this is actually, the reason this is actually really poignant to me is not only because it's a lot of data, but it poises us for really what we've been working for the past 2 years, actually, toward building a resource that just is not just for type 2 diabetes, but really encompasses common metabolic diseases. And many of you know there are existing knowledge portals that have been around for cardiovascular, cerebrovascular, and sleep disorders with other colleagues who have like-minded pursuits with us, and work to collaborate with those communities to build those portals. And really what this shows is that these communities are starting to come together to share their results, share their data, because many of these traits are relevant in so many other disease constraints. So this really sets us up for what we're going to talk about today. But I want to remind you, where we started with what the type 2 diabetes knowledge portal really was. It was truly based on a simple idea that propagates today and matters today even more, in my opinion, is that making genetic and genomic data more broadly accessible and useful really does hopefully have a significant impact on our ability to understand and treat human disease. And that's really where the portal started. It really started in understanding human genetics and making that data accessible. So the genetic data itself was really where we started, right? We had a motivated type 2 diabetes genetics community who wanted to share data. And I've said this many times, this is really important because this is a hallmark of what we're going to be doing going forward, both in a bigger community and also, more importantly, with new data types. So this was about genetic studies, GWAS, sequencing studies, exome chip, exome sequencing, whole exome. That's really what the root of what the portal was for the last 5 years. And the type 2 diabetes genetics community has been very successful in those types of studies because they've identified over now 500, I believe, 400 loci associated with type 2 diabetes. And we make these accessible on the portal. We also, represent the predicted type 2 diabetes effector genes also in the portal. So the portal really does a good job, I think, and has really sort of made its hallmark in representing genetic data, integrated, definitive results from the community, sort of the authoritative analysis protocols, the methods that are most valuable. And that's where we started with our first opportunity and challenge. But in order really to make sense of all of these genome-wide association study loci that we've identified, we need to figure out what the function is, what the direction of effect, what the magnitude, and moreover, what medicines we might be able to create from this knowledge. And so for that, we really need to, really leverage the many research areas that are needed in the function space. And this is not an easy process, and this is what many of our colleagues are working very hard to do. But our goal is to help bring those results and that data to the knowledge portal and integrate it with the genetic data over the coming years. And so for that, there's a new challenge, and that is going to require a change in our focus over the coming years, and that's really moving towards function. We really want to help people understand what is the variant, what is the regulatory effect, what tissue, what cell type, where do you go after, what is the gene if you have a locus, what is the gene that you should study in this region, what are the pathways, what are the mechanisms. These are a lot of activities that we'll be focusing on in the next couple of years. But in order to do that, you need data. Genetic data has really done a great job of creating foundations and making these publicly available to us in a consistent manner. But also there are other resources that are in the epigenomic space that have also come to maturity and are readily available for us to leverage that to understand the non-coding pieces of the genome, the tissue, the gene, the pathway. So for this, our goal is to catalog these results and integrate them into the knowledge portal framework, and then ultimately allow you to visualize these with the genetic data. So for that, there's new challenges, right? We need to understand what data types are the best, what data types are best to view chromosome accessibility, what data sets are for different cell types, tissues, single cell analyses and stuff like that. What information must be captured and retained and represented. For genetics, we're really good at that. We know how to document the methods, we know how to document the results, we know how to present the results. Now layering that with functional data is going to be a new opportunity for us. Also, we need to know what methods need to be run and how those need to be validated on the data to allow you to have access to those results. Then moreover, how you express relationships between these different types of data types, from genetic data to, peaks in a chromosome accessibility output or CRISPR screens. These types of data are all very different, but they all have relationships that we want to bring to bear within the knowledge portal. So our goal is to figure out how to represent these results and make them available in an open access framework. So our mission over the next couple of years, ideally, using the Type Diabetes Knowledge Portal as the cornerstone, will be to marry the best of the community-driven data and representation platform and software that we built for human genetics and leverage that for new data types, methods, and experimental approaches. And then also leverage the functional genomics community, which will be funded over these next 5 years, particularly for type diabetes, to really drive home the results from that, to really tackle all of those GWAS regions for type diabetes and its complications and represent those results in the portal. We also are very lucky because many of the other common metabolic disease communities are doing the exact same thing. So our goal is to create a fused platform to deliver the insights for both mechanism, magnitude, and markers for diabetes, effector genes, and common metabolic disease. So what are we doing to prepare? 4 things. So we aim to expand the data and software platform to respond to the massive data. As you saw from that last release, it's our biggest data push. That's very consistent with what we're seeing going forward, particularly as we expand to common metabolic diseases. So to do that, we hope to create a common resource for complex metabolic diseases and the traits while maintaining the community-driven content and contribution. That's really important. Really the reason that we are unique, I believe, from other large-scale resources that represent genetic data is that we have the communities that both think about the study design, execute the study, and write up the papers behind us, helping to supply the content and the documentation and the relevant interpretation of these results so that we can represent them on the portal. That's critical to preserve. But in order to sustain this massive amount of traits, data, methods, and tools, we need to erect a new framework that is efficient and intuitive, particularly because we have to represent and visualize new scientific results. And finally, our goal over the next couple months is to share, teach, and listen to our users as we roll this out in this new framework. So first, as I mentioned, new backend, excuse me. So this is not really a detail for today, but I want to just mention that was really important in allowing us to shift to a larger portal, one common resource that allows you to maintain all the other current portals, was that our backend used to be a MySQL-based database. We now have moved to a cloud-based Amazon storage place where we keep all our association statistics. This allows us to be able to scale and manage things like whole genome data that's going to be coming in. Moreover, also we converted to this new pipeline that allows us to store relationships between both the data that we store, that we bring in, and the methods that we run and the results of those, so that we can represent them very streamlined and very rapidly to you on the portal, both in a visualization or in APIs or in download if it's so needed. So this is a big change that allows us to run all of these methods that will be valuable for us as we load more GWAS data and also layer in annotations that are critical to analyze in concert. So that was a critical change. The other one was creating this platform to integrate the current type 2 diabetes, cardiovascular, cerebrovascular, and sleep disorder knowledge portal that have been around for many years and have a user base, but all are built on this exact same platform that I described to you. It was really challenging for people to go to different sites to find out about their favorite variant, their favorite gene, or their favorite trait, right? Also, the types of problems and challenges that we're working on in all of these different communities are largely similar. And so why not? And also it's very challenging to have 4 sites that you all need to maintain and make sure that they're fresh and always updating in content. So our goal was to really leverage the best of the expertise of the community, the ability for us to create integrated views of the data that cross all sorts of metabolic diseases, and also for us to have a more flexible and fast environment for you to look at these results. So just a little bit quick on the background of the Common Metabolic Disease Knowledge Portal. Our principles were the following: to maintain the community representation in the current portals. So make sure that those sites are still their own entity, their own hub, if you will, for the community. Therefore, we allow those existing URLs to still work for the current portals. And once we cut over to the new portal, they will cut over to the new framework and the exact same framework, same look and feel, that they currently have. And I'll show you that in a minute. These are all powered by that resource, the huge AMP resource, which is powered by the Type 2 diabetes software and platform that I mentioned to you earlier. And then we subsume this into a single resource that hosts all the data, methods, and features. Moreover, finally, to do this, this will engage, we hope, both a wider community of contributors and experts, but also a wider community of users. This will help us identify better data sets so we can aggregate, represent them for both genetic and genomic data for these traits on the portal. So this allowed us to bring together these communities, which you're probably all familiar with if you do these portals. But what's nice is each of these investigators and their colleagues are deeply engaged in building the common resource and also understanding how they're still stewards and representatives of their community by bringing more data, more traits, more concepts of what types of methods are most important, what types of data types are most important to our resource so that we're making it better as we go, which is really nice to have their continued engagement. So as of today, we have a beta version, which Maria is going to talk about, but this is sort of like just reminding you that this is like also a community-based effort, not just a software effort. So we're going to talk about the portal in a minute, but this is what it's going to look like. Now these 4 resources are all in one. But if you want to go to just the cardiovascular knowledge portal, when you type that URL in, once we convert over to the new framework, you will see the cardiovascular type diabetes knowledge portal if you just want to work in that framework. But you can also immediately bounce out and go to the common metabolic disease knowledge portal framework just by clicking here. Another feature that's important, because this allows the communities to still feel like they have their own hub, but also can work within a broader framework. So everything's analyzed, obviously, and represented on the same architecture, but it's just a matter of having different views based on your preference. So example, this is the different banners you might see in different modules as you navigate through the site. But then finally, what's critical is all these data sets are now available for you to query in one place. This is now 130 data sets over 269 traits. And that's just the beginning. But what's beautiful about this is that it's the same framework you're used to with the Type 2 diabetes knowledge portal, where you can filter data sets by group, type, phenotype. All the detailed documentation and descriptions are still there. So you can drill down, you can learn about your favorite disease, data set, all the information about it, how it's constructed, the publication. If it's available for download, you can get it here, and all the information about, specific content and analysis protocols and how the study design was made. And then finally, what we're hoping to do here is to enhance this with a new reasoned ontology, because you can imagine if we have over 270 traits, we're going to get up to 300 really fast. We need an ontology that resonates both with geneticists, clinicians, and the broader user community who's going to come at this from a basic biology standpoint. So we've been thinking about ontologies that will resonate there, and we're going to be mapping them to classic styles, so Mondo and EFO, but we'll also be building it in a way that is sort of persona-based, we think, in terms of how we represent these traits, so that the communities, if you're in each community will have their own sort of lens within their community portals, but also in the broader resource will have a reasoned ontology that you can navigate. As you see, we have one currently, but I think we're going to have to do a better job of this as we go forward. Moreover, we plan to have more detailed phenotype information for every single phenotype that we capture in the portal, how it was defined by the community and other things that it maps to, so you can understand where it came from. And one thing that is really critical is that we've maintained provenance, meaning when a community has definitively said, this is our data set that we want to represent, on the portal, we also maintain, so you can have reference on the common metabolic disease framework, what portal or what community it came from. So this helps us, see as the resource grows, the fact that there's all this commitment and investment of these other communities coming to build a common resource. Then finally, now that you have all this data, we need to build a new framework. It's a great timing. The portal's about 5 years old. We're about to be in our hopeful renewal for the next 5 years. It was a great time to erect a new framework. And the principles behind this framework are that we want it to be efficient and intuitive, in both representing and visualizing scientific results. So a couple things on that before Maria dives in. This is a very exciting thing for us. And many of you who have used the portal have noticed that it's a little slow. A little slow is a kind word. You know, when you log in to click SLC38 is your favorite gene, it takes a while to even load the gene page. And we don't want that anymore. I think that, you know, we have our ability to spend some time and really invest in efficient software to make the site come up fast, efficiently. So that's really what our engineers have been working on, and it's really quite awesome to see what they've done. And additionally, this is a little preview, but it's way faster. The page loads is right away. You're going to see the information. The other thing that you may have noticed with the old portal is that not all the page was using space. We have this beautiful design team led by DK, who DK Zhang is probably on the call, that has full page views now. I know that, you know, that may be simple, but it really does. It has a much sleeker and cleaner look. Additionally, we've also integrated a card-based construction. So what I mean by that is each section is a card. And the reason for that is it's, you know, at one, linear in terms of its, you know, the way it flows. It also makes it a very modular and flexible design. If we have a new feature, we can pop it in wherever we think it's appropriate in the workflow. And it also is just a cleaner way of representing the results. And finally, this is more something that will be growing over time, which is, I would call it, maybe this is not the right design term, but for me it is, it's an iterative workflow in the sense that as you're on the page, as you go down and you do things on the page, the bottom of the page, and this is a common thing that's done in URLs, you know, adapts to whatever you're clicking so that your workflow stays with you. So for example, if you're changing your traits up here, everything below that fold changes based on what you did above so that you can work in a workflow environment. And Maria's going to show you some of that workflow next. I think this is a very nice way of, you know, shifting us to a new framework that'll be both nimble and faster, and hopefully you all like it. Now, what you're also going to see are different ways of viewing things. Some things may not look the same. Some things are going to come up in sort of a different, you know, presentation, as you might already see from the phenotypes. Really, what's going to be important to us the coming months is for you to give us feedback about that. What do you like? What's missing? What do you want more of? What's really working? And so Maria's going to talk about ways that you can give us feedback right away. But as always, just email us. You know, we are, at the end of the day, you know, still all colleagues and researchers working together. So a simple email is really easy too. So with that, I'm going to turn it over to Maria, who's going to walk you through a live demo. And then I'll have a couple of comments at the end. Maria, do you want me to stop share?
Yeah. Okay. Now I'm sharing my screen. And let's see. All right, you should all be able to see my screen, I hope, with a web page.
Yep.
Okay, so this is our Common Metabolic Diseases Knowledge Portal beta version. I want to stress that it is really still under construction. Not everything that will be there is there yet. Not all of the help and explanatory text that will be there is there at this point. And some of the visualizations are still being worked on and will change. But it all works, and we would love it if you would explore it and give us feedback. And here on the home page, there's a link where we just put a feedback form to make it really easy for you to do that. But as Noelle said, you know, just emailing us is always fine too. Okay, so there. Oh yeah, and I also wanted to say I want to thank our engineers so much because I'm so thrilled to be giving a live demo. It hasn't really been possible with the current portal, and as you'll see, this one is really nice and fast, so it's fantastic. So there are maybe about 5 major components of this redesigned site. Actually, they're still in flux, but some of the major ones are the region page, the variant page, the gene page, the phenotype page, and a new kind of page that we're calling the dataset inspector that I'll talk about a little bit later. So today I'm just going to go briefly over 2 of these pages, the region page and the phenotype page, and just give you an idea of what's on them. Let me see. So I'm going to start with the region page. I'm going to copy and paste a region in here. Yes, already you can see how fast this is coming up. It's not up yet, but it's there we go. Okay, and so the purpose of this page, as we see it, is we think most people who are coming to this page will have a genetic association locus that's significantly associated with a phenotype or disease that they are interested in. And so they're going to want to come to this page and get a quick view of what is known about this region. And we want to start out with presenting a quick summary, a real overview of what's going on in the region. and then allow people to drill down to the fine details if they really want to know, at the level of individual variants or anything else. So I'm just going to quickly take you through this page. So first of all, this shows the region you're on, but if you decide you want to change the extent of the region, you can easily edit it here. You can also just go to a gene page by typing in a gene name here. And as Noelle said, the page is set for a particular phenotype in general. This section here summarizes all phenotypes, but then everything below pertains to the phenotype that is shown here. And if you want to change that, you can do that easily by just typing in something else, and you'll get a selection of the phenotypes that match that string. And then if you hit return, you will set the phenotype for the page to that phenotype. Okay, so this next section here, this next card, is meant to represent an overview of the phenotypes for which there are significant associations in this region. And we're still playing with this visualization. We're really interested in your feedback on what's a useful way to look at this. And we actually have two alternative visualizations now that I'll show. So here's one of them. This bar graph shows the phenotypes for which there are genome-wide significant associations in the region. So this, and also they're color-coded by their phenotype group, so glycemic phenotypes are purple and so forth. And then the less significant associations are shown here. This view looks a lot like it does on the current portal with the bubbles. And if view more traits would, yeah, that's not even all of them. You see what we mean when we say we have a lot of phenotypes, and we have to find clear ways to display this, because obviously this is just overwhelming. So this is one potential display for this. Another way to look at it is this, I think, very cool looking graphic. Now this collapses the phenotypes into their groups, these are groups that we have set at the portal, and we are definitely planning to edit these and possibly make them into an ontology. Right now it's just a hierarchy. But the way this works is that each of these bars represents all the phenotypes in that group for which there are genome-wide significant associations. And you can expand a group and see each of the individual phenotypes and see the p-value for them. And this reminds me that I forgot to say, all of the p-values on this page represent bottom line meta-analyzed p-values. So we perform meta-analysis that's overlap aware for sample overlap between data sets. This is a method worked out by our colleagues at University of Michigan. And so we do this so that for each data set, for each phenotype, we come up with the bottom line, sort of consensus or best p-value for that phenotype. So this takes a lot of the, it kind of collapses the associations to the consensus value. And I'll talk a little bit more about that later, why you still might want to see p-values from individual data sets, of course. Anyway, so this is another display here, and we really will be interested to hear whether you like this display, something in between. We're really interested in your feedback. Okay, next is a locus zoom plot, that you're all very familiar with genes below it. And this is not complete yet. For one thing, this make LD reference does not work yet, but it will. And then in the current portal, we have epigenomic tracks below this browser, and they will reappear here. And we're also planning to make a lot more tracks available, not just the chromatin states and a little bit of ATAC-seq data that we have now, but much more in different data types. Next, we have a table at the top, associations. Again, these are the bottom line p-values. And the links will take you to either a region page encompassing the region of this variant, or it'll take you to the variant page. We will put in the gene names here rather than the ensemble IDs. That's coming. And so, in addition to everything I've showed you here, we're going to add a module to the page that we're calling the variant prioritizer, and I'll show some screenshots of that, or mock-ups of that a little bit later. At this point, maybe I should stop and see if there are any questions or comments about the region page before I go on to the phenotype page.
I'm gonna try and see it work. No open questions. All
Okay.
right,
Well, let's go on.
let's go.
Okay, so to the next type of page I want to talk about is the phenotype page. And first of all, I want to show you what are the phenotype page as such is like in the current portal. So we have a page for each phenotype that has an interactive Manhattan plot. For one thing, it's not a true Manhattan plot. A lot of people have commented on this. For example, this one starts at p times e to the minus 60. So this is like the very tips of the skyscrapers. It's not what people are used to seeing. And there's a table at the top, hits below it. But the main thing here is that we have so many data sets. If you're not intimately familiar with these data sets, it's really hard to know which one you want to look at, which one is going to give you the most representative, definitive Manhattan plot for the phenotype. That's pretty impossible to know. So that's one difficulty with this that we're trying to address. Another type of information that we have that's relative to a particular phenotype is a list of predicted effector genes. And right now in production, all we have is one list for type 2 diabetes. But we're working with other collaborators some of whom are doing this type of prediction for multiple phenotypes. So there are going to be a lot of these, and they properly belong in with information about that phenotype. So this and the Manhattan plot are two disparate things that really should be together. So let me show you the phenotype page here. So you can get to the phenotype page either from the home page, or you can, anytime you mouse over a phenotype here, you can either, so let me just show this. You can set the phenotype, that changes the phenotype for the rest of the page. But then if you want to go to the phenotype page for that phenotype, that takes you there. So here is the phenotype page. And again, it's all based on bottom line p-values. So at the top we have a Manhattan plot, and we're also showing a QQ plot, which is new. We don't have that in the current portal. And here we have a table of the top association, the top bottom line associations across the genome for this phenotype that we've selected. And I should have said before, all these tables will be filterable also. So you can, even though they have multiple pages, you can find a variant that you're looking for. Okay, now the bottom line p-values, as I said, are made from meta-analysis. But you probably want to know which data sets went into that meta-analysis. That's really relevant. So this table will show you that. These names will be, these are our internal names, and the final portal will show the names, the user-friendly names that you already see on the portal. But this list tells you which data sets went into that analysis and how big they are, which is really relevant for deciding how much you want to believe the bottom line p-value.
And then just one comment, Maria, sorry to interrupt, but what's nice about this is that this will also be, I believe, we were hoping this would be clickable and allow you to go into detailed information about that particular data set. So we know that people really want to be able to still drill down into their individual data sets, particularly if they're contributed by a community. This function will enable you to do that, but also have the full genetic data rolled up for you, as Maria said, the bottom line. Really the key is here, I think having all these data sets is really great for an expert geneticist who really wants to understand in different data sets, and they understand that, small data sets, you take them with a certain grain of salt, bigger data sets, and so on and so forth. But a basic biologist coming at this site, if we gave them all these data sets, they'd be like, what? But that's why the bottom line to us is really the way to sort of say, marquee finding for this phenotype is this. But for you, if you want to really understand the information, it's all still here, just presented to you in a little bit more distilled format. So that's kind of really the difference that I think is a nice shift from our current portal to the new framework. Sorry, Maria, continue.
That's okay. Also on this phenotype page right now is a table of our Grigor results. And so if you're interested in the phenotype, you probably want to know which tissues may be relevant to that phenotype. And so Grigor takes cell type specific annotations well, some of them are listed here, chromatin states, DNA's hypersensitivity, and so forth. And it integrates those with meta-analyzed p-values or for genetic associations, and then gives a value for enrichment of the tissue-specific annotations in genetically associated regions. So this will, for example, here we're looking at a type 2 diabetes phenotype, and we've got our top tissues as being pancreatic tissues, which makes sense. Other things that will be on this page in the future, as I said, effector gene lists. If this is a phenotype for which there is a list of predicted effector genes, it'll be on this page. There will be other phenotype-specific information, such as a definition, links to ontology terms, and so forth. And finally, we need to give users a way to get an overview of all the phenotypes that are in the portal. There are so many phenotypes, you might not even know what's in there. So we definitely are going to make a page that lets people browse and search the entire complement of phenotypes to see what's there. Okay, let's see, before I finish the live demo, I wanted to just illustrate what Noelle told us about how to get to the disease-specific portals, because it's just cool to see how well it works from the homepage. So here is the CMD KP homepage, and if you want to explore by disease area, for example, you can just click that, and that takes you to the familiar portal for your disease community. And this will have the phenotypes and data sets that are specific to that disease. And at any time, if you want to look at everything together, you can go back to the CMD KP. All right, now I'm going to switch back to the PowerPoint. Let's see. Okay. So I mentioned the dataset inspector page. So the idea is that, yes, while bottom line p-values are really valuable for giving an overview, it's also very valuable to be able to see associations in a particular data set, particularly if say you've done a study and you want to see if your top associations are replicated in a certain data set. You really do want to see that data set rather than bottom line. So this is just a wireframe, just like an outline of what will be on this page, which is not made yet. And so it will be particular to a data set. And so it will have the description. As you know, we already have detailed descriptions of the data sets, like, the publications they came from, the cohorts that are in them, and so forth. That will all be there. And it will also list the phenotypes that were analyzed in that data set. And then you'll be able to look at a Manhattan plot from that data set alone for each of the phenotypes that were analyzed, and also a table of those top variants. Also not shown here, but we do, when we use these data sets in various GWAS post-processing methods, we will show here which methods have been applied to this data set in particular. Okay, and now I'm going to talk a little bit about a future workflow, which we are calling the variant prioritizer. It will probably be on the region page. And the idea here is that is moving from variant to function. That you want to go from a GWAS, a genetic association signal, to figuring out which gene is actually affected and what it is doing in the context of disease. So this is a way to help people move along that track. And this is all really vague at this point, and we would love your feedback again on this. So we visualize it starting with a genome browser. This is LocusZoom. It's a good candidate for that. We'll see if it can work for this type of thing. But so we would allow users to select tracks of interest to display under it, and not only these tracks, which are the ones that we currently show in the portal, but lots of tracks with different data types. So people could really zoom in and look in detail and see which variants or sets of variants were of most interest. And here's another view of that. This is a visualization that the patterns group at the Broad has been working on with us. Leah and Noam and Bang have designed this. And so here's part of it. You could think of this as a variant selector. There's a space where you can select variants from, you can just select a whole credible set, or you can select individual variants or combinations of them. And then you can also select genes that you want to look at, tissues that are of interest, and lines of evidence like epigenomic marks, or the activity by contact method, which identifies enhancers from Jesse Ingritz's lab. So you'd be able to select all the things that you were most interested in, and then see a visualization that summarizes all this and displays it. So for example, I know this is kind of hard to see, but these are variants going across the top, and there's some tissues going down here, and the colors indicate the line of evidence and the tissue that are associated with those variants. So this is an in-progress idea that we have that might help people prioritize variants. And then finally, a lot of people would want to just get all the data. And so we want to make it easy for them too. We could call this the extractor, where you can upload a set of variants of interest. You can see a list of the available data types and methods and just check off whatever you want, and then download the whole thing and have the file, and do your own analysis. So that's also in progress in our group. And I think that is my last slide. And so I'll turn it, well, first of all, are there any questions or comments about what I've shown?
Well, people are potentially popping into the Q&A session. Just a comment about the variant prioritizer. I think this is, we hope to be one of the crown jewels of the workflows that we present in the portal, because this really does bring together every piece of relevant information, right? So it's challenging because the first thing you really want, I think one of the places we think we're starting is a visualizer, which is just the browser of the genome, some sort of linear space where you're seeing a region, you're seeing, the genes involved in the region, some genetic information, and the relevant, thank you, Maria, the relevant tissues, right? So start with that. But really, what this is about for people is they have a region, they have a set of genes. They want to know what data sets are available to help them choose between which gene or which variant. And so we felt that the visualization is the first place to start. And you can see this as like a layering approach, where you would start with the visualizer in a genome browser, and then allow yourself to add these tracks because you want to customize them. You want to customize what cell types you want to see, what tissues you want to see, what data types or approaches you want to see. And then in the visualizer itself, which we have a little bit pictured up here a bit at the top, the browser would actually add those tracks to the visualizer so you could get a nice picture of it. Then let's say you actually want to drill down and get the results. The next visualization kind of shows that drill down. So I think many of you might have seen this before. We call this the variant focus table. It's sort of the same expression of that, same results and things like that. And remember, this is all data that's taken from our sister node at the Diabetes Epigenome Atlas. So all that data that's housed there, the raw data, we're finding now a way to represent the distilled, processed results of that, the peaks and the relevant annotations, representations in a visualizer. So that's the goal of this. And then, moreover, you need to be able to do something with that, ideally, right? You need to see the results. Which results should you use? We don't know which ones to trust more than others at this point, right, as a field, but our goal is to present the information to you so that you can at least have access to it. And then I think the third page, which is the sort of the extractor, as Maria referenced, that is about really for someone like Jesse Ingritz, who really wants to extract all those annotations, the sort of the matrix, and run his own method, or wants to do this for multiple regions of the genome. We really want to make it a computational approach as well as a visualization, a tool, and a computational approach. So that's kind of the process with that. And I think this is something that we're going to be working over time. Oh, we have a question. Excellent. What was the question? Are link RNAs included for analysis? Good question. Not at this point, but we can absolutely figure out how to get those data sets in. I think that's a good suggestion. I don't know if actually, it's a good question for potentially for DGA if they're using those data sets yet. Good question. What else? So let's move I will actually follow up on that question for you because we don't have the answer right away. Let me go to the next slide. Maria, you can just use your slide maybe.
Would you like me to stop sharing, or shall I just do the slides?
I'll share.
Okay, I'll stop sharing.
Okay. All right, lovely. So with that, just a couple sort of, so one thing Maria did not reference as well, which you may be used to seeing as well as I'm sharing my screen. Hold on, I'll just talk over that, is the variant page, for example, and the gene page. Those are all going to come online shortly. Versions of them are already available if you're poking around. And those all will exist on the new framework. We just haven't fully developed them yet. So if you're asking about that, those are coming online. And those will be slightly different in terms of what they represent, but the variant page will still have things like the PWAS that you're used to seeing. And also, variant across all the traits will also be there as well. And also the gene page will be envisioned in a different way because we really want that to be about the gene, not about the region. So that has a slightly different approach that we'll be taking over time. But in wrapping up, just information broadly about the resource and other things that are going on, which I think will be relevant to this group. All of the other portals. So now that, the 4 portals that are for common metabolic diseases will be coming together. We also have, the platform that powers this. We are building a website that sort of, gives you all the information relevant to this. And so we've coined the name Human Genetics Amplifier, which is the platform that I was mentioning earlier that powers the entire data, software, and UI of the portals. And we're going to pilot this. This currently exists as our KP for CDorg, and we'll be restyling this over the coming months. What's important is you can get to all the other portals as well here too, and find out information about the overall resource as well here. But I thought this might be relevant to this group, which is that there are other portals that are outside the common metabolic disease framework that we've been very lucky with other communities because of the resource and the work that the AMP partnership has done to build this portal. Many communities have really liked what we've done, and it's pretty awesome. And really what's been obviously nice as well in learning from these new communities is they have different ways of thinking about things and visualizing and wanting to bring in more data. So for this, in March, we launched the musculoskeletal knowledge portal. And it brings together the bone communities, bone disease and musculoskeletal disease community. And we already have over 1,500 users of this portal. So that's pretty cool. And just this release, the osteoarthritis community came to the collaboration as well. So those of you who know Ellie Zaghini from the type 2 diabetes world, she has been a contributor to the musculoskeletal knowledge portal and will be helping us, build this as well going forward, which has been really great because this has been a grassroots effort that also has produced another portal. Also coming, hopefully this spring, is a lung disease knowledge portal. So this will bring together COPD, asthma, and interstitial lung disease results. And this is a portal that we've been working on in the background as well for several months, actually a year now, and hoping to launch later. We want to make sure we launch it with all relevant lung disease data sets. And what's really useful about this is, I think, again, this is a common complex set of diseases. Again, same platform, really AMP enabled this. But what's really nice about this, I think, is that a lot of the, and the musculoskeletal, a lot of the cohorts, the large biobanks, the top meds, the MVPs have all these phenotypes. And so, this is really forcing us as the development group and the data team to think about how do we leverage those large scale resources? How do we leverage top med? How do we leverage MVP, UK Biobank? And that's been, it's a slow process because they are their own entity and they have these working groups and very complex ways of working. But I think this will help us sort of open the door to ways of representing their results, hopefully in a way that, honors every sort of contributor and community. So that's really where that's headed. Thinking about the common metabolic disease knowledge portal, the NIDDK's commitment to diabetes is awesome. And they helped us with supplemental funding with Kyle Galton and Steve Rich to develop a type 1 diabetes knowledge portal that we'll be launching hopefully this summer as well with the common metabolic knowledge portal. This is coming along nicely. We already have data loaded both from Kyle and Steve. This has been really wonderful to work with these people who we've worked with many years in the type 2 diabetes space. Now learning about type 1 is a very different beast. We also have to figure out how we represent the HLA region and things like that. So it's a very different disease from a biology perspective. So we'll have some special tweaks there, but look for that soon. That will be coming online as a beta release in June. So upcoming webinars, we plan to share more of this with you all. Again, on Thursdays at noon. The next one will be July 16th. That one ought to be fun. We hope to have the type 1 diabetes out and about circulating, and a more advanced version of the new framework. Again, type 1 will also be on the new framework. I'm going to talk about rollout now. This is important. So the beta site is out. As of today, you can play around with it. You as lucky audience participants get a sneak peek at it. Remember, it's under active development. You might see something, break. So be patient. You might see something be added that wasn't there yesterday and is there now over the next 2 months. That's what's going to happen with that. All the current sites that you're used to dealing with are available and open access to you still, and that has not changed. We're planning to have a live launch and data freeze somewhere around July 30th of the new portal. So this means that there'll be data parity across all the current cardiometabolic portals with the new framework, and that they'll be running in parallel for a couple weeks as we stabilize, make sure everything's working, correct all the data, which we're doing as we go, but adding content, really polishing it up, making sure it's working well. We're aiming a phase out. That sounds a little scary, but it's really not. I don't think around August 15th. And at that point, the current URLs that you're used to for the old framework will redirect to the new one. And the new site will be what we update going forward. But during this time, we'll be doing feedback and training. If you're interested in the training session for your lab, if you're interested in the feature, if you're interested in just giving us straight up feedback, please do so. I think we should share the feedback form. I don't know if Maria showed that or not. I popped out for a second, but we have a feedback form that we can show. I think, Maria, do you want to show that before we break? I know there's some questions too.
I did show it actually. Yeah, it's a link that's right on the homepage.
Beautiful. So that's kind of our plan. And again, we want your feedback. This is the team that we are very lucky to work with, who helped build this, particularly these engineers who really rethought the entire framework from scratch, building it from the ground up for this new launch, which is pretty awesome. Again, our wonderful wider collaborators from Michigan and EBI and UCSD that work with us to build this overall human genetics amplifier platform for AMP type diabetes that we're really blessed to work with. And finally, the AMP partnership, with which the data contributors, the thoughts, the ideas would not be possible. So again, thank you. And I'm going to pop to the questions, and people can add a couple others. Oh, fantastic news. Thanks, Phil. That's awesome. Any information about the correlation between different traits is available in the portal? That's a good question. No, that's not currently available in terms of the correlation. You can look at results for multiple traits layered. So you could say, like, show me all the variants associated with type 2 diabetes at this p-value and cardiovascular disease. A correlation, no, we do not have those results. We're going to do some of that work in the coming months. You should see that now that we actually have the common metabolic resource with all the data together, we actually have an ability to do that at scale. So that'll be coming. Any other questions or comments? Again, please use the site. I think that's the best way. For many of us, we spent a lot of time with a smaller group of people, who are our contributors, giving us feedback. But what I love about, having a larger user base is that people think about things from, I want to use it, not I'm contributing data. I think having that sort of flipped around will be really lovely for us. So, if you have any feedback, please give it to us. We're excited to hear it. So with that, I think unless there's other questions, I will say thank you, and we'll see you in July. And hopefully people's labs will start to slowly go back to work, and hopefully everyone will stay safe and healthy between that time. Thank you for joining us. Take care.
Thank you. Bye-bye.